All right, welcome to Chapter 26, Fluid Electrolyte Balance and Acid-Base Balance. We will jump right in and begin talking about your bodily fluid compartments. So you want to know that there are two of them, intracellular fluid and extracellular fluid, and then know that the extracellular fluid is divided between the plasma, so extracellular fluid in your bloodstream, and interstitial fluid, which is all of the other little in-between places in the body, which also includes things like lymph and cerebral spinal fluid, synovial fluid, and whatnot. What you'll notice is missing is whatever is in your bladder and whatever is in your stomach and gastrointestinal tract. So all of those compartments, your GI tract, your urogenital tract, and your respiratory tract, those are considered to be parts of the outside world that sort of your body is surrounding. So the fluid in your intestines doesn't count as part of your bodily fluids. You also want to know their relative proportions. So you want to know intracellular fluid is the biggest compartment, followed by interstitial, then plasma, and all of those other things we were talking about, like lymph and serous fluids. And then when it comes to fluids, you're mostly talking about water. Water is the primary component in all of the fluids of your body. And you just want to keep in mind that for the most part, water moves by osmosis. Hydrostatic pressure plays a role, but it's mostly osmosis. And your body can't control where the water goes. Right? So what your body does is pump solutes from one compartment to another compartment so that the water will follow by osmosis. So your body uses solutes, primarily sodium, to direct the water from one bodily compartment to another. When it comes to the different intracellular fluids, or the different bodily fluids, what you want to know for each one is basically the major cation and the major anion. So you will notice interstitial fluid and plasma are very similar in that they're both very high in sodium, right? Because of our friend the sodium potassium ATPase. It's pumping sodium outside of the cells. So both of your extracellular fluids are going to be high in sodium. And then they have a fair amount of chlorine for charge balance. And there's our friend bicarbonate, which you are going to see plays a role in maintaining pH. I wouldn't worry about very little versus very much protein, but it is interesting to think about if you want to get into the weeds of osmotic balance, which we don't, so we'll move on. Then you have intracellular fluid. Here it is low sodium. So the major cation is potassium, and the major anion, you can just call this a phosphate. I think it's like hypophosphate. It plays a role in maintaining pH, but we kind of gloss over that. I am going to want you to remember that there's a fair amount of protein in your intracellular fluid, because we're going to talk about that when we're talking about the maintenance of intracellular pH. So then when it comes to overall electrolyte balance, some books and our book talks a little bit about how the different electrolytes are balanced. I just lump it all together in this one slide here, and say that electrolyte balance is important. And the two biggest reasons why are these two right here. So first of all, solutes like sodium and potassium are used to maintain water balance. And then, can't speak today, electrolytes like sodium, potassium, and calcium all play a vital role in all of your excitable tissues. So your nerves, your smooth muscle, your cardiac muscle, your skeletal muscle, none of that would work well if you had too much or too little of your electrolytes on one side or another of the membrane. So know that anytime your electrolytes are screwed up, that can mess up your muscles and your nervous system. And sodium is there for water balance. So now we're on sodium. What you want to know about it is that in addition to playing a role in electrical excitability and all of that stuff, this is your body's most important regulator of water balance. The most powerful control, the most important control mechanism that your body has over sodium balance is aldosterone levels. Um, and aldosterone levels are affected by, if you remember, there's actually four things. But for this chapter, it just focuses on two. It's potassium levels and blood pressure or volume. Um, if you look at the chart here, this just shows, I don't know why this has a whole thing for um, blood sodium levels do not affect potassium, but what you want to remember is that, blood sodium levels do not affect sodium. But you want to remember that because you have the sodium potassium ATPase in your bloodstream, potassium and sodium cannot be balanced off one another. They can't be independently regulated. So your sodium levels are going to fluctuate to maintain blood pressure and to maintain your potassium levels. But if things get out of whack, if you've got high potassium and high sodium, then it's hard to maintain proper blood pressure, right? Because as your potassium levels rise, what your kidneys do is hold on to more sodium. So more sodium stays into the bloodstream and potassium gets kicked into the urine. But if you've got high potassium and high sodium, then you can decrease your potassium levels, but your sodium levels are always going to be too high, and that's going to raise your blood pressure. And so this is why we're told to watch our sodium intake so that that doesn't get too high. Next up is water intake. There are three important things I want you to notice here. One is that the factors that lead to the stimulation of your thirst mechanism. So increased plasma osmolarity and the sensation of dry mouth are two of the most important ones. And I want you to know that the thirst centers, the part of your brain that makes you aware of the fact that you are thirsty or should drink water, is located in your hypothalamus. Then the other thing I want you to know is how the negative feedback mechanisms work. And the important thing to note here is that the first point at which you get negative feedback is when your mouth is no longer dry and when your stomach starts to distend. So this tells the body early on that you're drinking water and starts to shut off the thirst centers in your hypothalamus. If you kept drinking until the water got into your bloodstream and that now more dilute blood got up to your hypothalamus, you would drink way too much water. So having the water in the mouth and distension of the stomach shutting off the thirst centers ensures that you don't overhydrate. So now this is sort of the meat of the chapter, is acid-base balance. It can get complicated, but we are going to make it as simple as possible. So first up, just know that normal blood pH is about 7.35 to 7.4. And I won't talk too much about that. Then, the other thing you want to know is that generally speaking, if you increase your metabolism, this lowers the pH of your tissues. And I won't ask you about all of these conditions, but it's nice to think about so that you just sort of understand. Some of these things we know already, right? Your muscles churn out lactic acid. When you go anaerobic, that's going to decrease your blood pH. You already know that if you have higher amounts of CO2 dissolved into the plasma, that's going to get converted into carbonic acid. So this really should say H2CO3. but it doesn't matter. Carbonic acid from CO2 is going to lower your blood pH. So your body has mechanisms in place to maintain blood pH. You have chemical buffer systems and physiological buffer systems. The physiological buffer systems we've already gone over. That's your respiratory response and your kidneys response in managing CO2 and hydrogen bicarbonate levels. We also have these chemical buffer systems, which keep your pH stable. Um, so a buffer is any group of chemicals or chemical that you add to a solution that keeps the pH stable. So it'll neutralize acids or bases. The buffer systems that your bodies work are all composed of a weak acid and a weak base. So before we go into those details, I just want you to understand the difference between strong and weak. So here we have two acids. Hydrochloric acid is a strong acid. When you put a whole bunch of molecules of hydrogen chloride into solution, they all dissociate into hydrogen and chloride ions. When you take a whole bunch of carbonic acid molecules and dump them in water, only some portion of the carbonic acid dissolves and liberates those free hydrogen ions. So you've got a reserve of acid still floating around undissolved in the bloodstream. That is a weak acid. And a weak base would work the same way. It doesn't fully dissolve and liberate ions that can then interact with hydrogen ions and pull them out of solution. So as we said before, oh whoops, sorry, this is the example I'm giving here. So we're still talking about carbonic acid as a weak acid. What that means is that it can neutralize a strong base. So here's your carbonic acid. When you dump it in water, a certain amount of it falls apart, but you've still got also a certain amount of it intact. It's sort of some acid in reserve. So if you then add to this solution a strong base like sodium hydroxide, this immediately dissolves completely into sodium ions and hydroxide ions. And if you look here, here's a positive, here's a negative. These two ions love to get together and form a molecule of water, and then these two form sodium bicarbonate. I think I drew that correctly. So when you add this strong base, it is neutralized by this weak acid. So what's going to happen is when these two reactions happen here, the formation of water and sodium bicarbonate, that means you have less of this and that. So more of this is then going to fall apart, because there's always going to be in solution the same ratio of intact carbonic acid to dissolved carbonic acid. So the dissolved carbonic acid neutralizes the base, and then the undissolved carbonic acid dissolves to replace the hydrogen ions that you lost here. So you can add base to the solution and the pH doesn't change because you have excess acid that has not liberated its hydrogen ions. You can think of weak acids as spare hydrogen ions that can be released when needed. So then we go into the chemical buffer systems. The truth of the chemical buffer systems is a little bit more complicated than what I'm going to tell you. Just to keep things simple, what we're going to do is put each of the three buffer systems primarily in one fluid compartment. It's usually for your intracellular fluid and your plasma, there are multiple buffering systems there. But we're going to do one buffering system, or we're going to put each buffering system in just one fluid. So first up is the bicarbonate buffer system. You want to know what your weak acid and what your weak base is. So it's carbonic acid and sodium bicarbonate. Don't worry about recognizing the chemical structures, just know the name carbonic acid for your weak acid, sodium bicarbonate for your weak base, and then know that this is your most important extracellular fluid buffer. So both your plasma and your interstitial fluid are going to use bicarbonate. And if we go back, right, here's your bicarbonate ion. And there's always lots of sodium, so sodium bicarbonate and carbonic acid are always going to be in high abundance in extracellular fluids. So if you look here, this is your weak acid. This is actually your weak base here. Um, so these two things kind of balance each other out. The carbonic acid dissolves and neutralizes bases, and the sodium bicarbonate will dissolve and neutralize acids. But you don't need to understand all of the chemistry. You will not see these chemical reactions on any exam in my classroom. Then you have the phosphate buffer system, which is similar to the carbonic acid or bicarbonate buffer system. Don't worry about dihydrogen versus monohydrogen phosphate. These two chemical symbols mean nothing to us, and the words mean almost less. So just know that your phosphate buffer system has a weak acid and a weak base that both have phosphates in them, and that this is what it is used in your urine to keep your urine from becoming too acidic or too basic. The reason why we have this buffer system active in the urine is because you recall what your kidney does is keep your blood's pH balanced by dumping excess hydrogen ions or excess bicarbonate ions into the urine. So the amount of bicarbonate in your urine is always going to fluctuate to suit the pH needs of the bloodstream. So you have to have a different buffering system to counteract that, because your urine can't just let a bicarbonate buffer system run by itself, because it's always going to be being perturbed by your bloodstream. Then we have the protein buffering system. This is one of my favorite topics. I'm going to geek out on you a little bit, and we go kind of full circle to Anatomy and Physiology 1 and talk about proteins. So the protein buffering system is the primary buffering system in intracellular fluid. You also do have phosphates in your intracellular fluid, but we're going to say protein, intracellular fluid. And the way it works is that, whoops, I went too far. All of your proteins, oh wait, I thought I had deleted this one, ignore that. These are from the old textbook and they're copyright protected. So these are from our current textbook. So the way proteins can work as buffers is that every one of your proteins has a number of amino acid side chains like you find on aspartic acid or lysine that can act as either acids or bases. So aspartic acid or aspartate has this COOH group on it. It's called carboxylic acid. Under the right conditions, this carbon right here can let go of that hydrogen. So instead of COH, it's CO negative with a liberated hydrogen ion. So this amino acid side group can release hydrogen ions into solution, which lowers the pH, so it acts like an acid. This side group here, this amino group, NH2, under normal circumstances is just NH2. But if the pH goes down, hydrogen ion concentration goes up, it has the ability to absorb a hydrogen ion and become NH3. So you'll notice what happens here is that this amino acid side group is now taking on a negative charge, and this amino acid side group is now taking on a positive charge. This is why acids and bases, but acids are better at it, can denature proteins because it changes your charge distribution, right? So here we have a protein. I forget what protein this is supposed to be. Go to this Wikipedia page, it'll tell you, oh, it's albumin from egg white. So here we have, this is that aspartic acid side group with the negative charge on it now. So it's released its hydrogen ion. It's negatively charged, which means it's polar, so it's on the outside of the protein where it can hydrogen bond with water. And then here are all of our lysines, right? Our NH2s are all tucked into the middle of the protein because they don't have any charge on them. They can't hydrogen bond, so they're all hydrophobic. So you have hydrophilic amino acid side groups exposed to the water on the outside of the protein, hydrophilic on the outside, hydrophobic on the inside. If I add an acid to this solution, right, well then these carboxylic acid groups, if there's more acid, they are going to be more likely to pick that hydrogen ion back up. They then lose their charge, and if these amino side groups on the lysine end up picking up a hydrogen ion, they are now positively charged. So what was the hydrophobic regions of the protein, excuse me, I keep on doing that, on the outside, what were hydrophilic amino acid side groups become hydrophobic, and our hydrophobic amino groups, which were on the inside, become hydrophilic. And when you rearrange the charges, that just causes the protein to unfold because now all the hydrophobic hydrophilic interactions are all out of whack. And this is why proteins denature when you change the pH on them, because you're changing the charges on all of those acidic or basic amino acid side groups. Isn't that interesting? Doesn't that just make you feel so much better about all that time you put into learning protein structure in Anatomy and Physiology 1? That's one of my favorite parts of the class, is that slide right there. What else do we want to know? Sorry. So the chemical buffering systems you want to know work really fast, right? So chemistry happens really quickly, right? But you only have so much bicarbonate or so many proteins or so much phosphate in a bodily fluid, so they have a limited capacity. Under normal circumstances, they work well enough to keep the pH from changing. But if there's too much acid or too much base, too big of a change in the physiology, then they can't keep up. And that's my email getting in the way there. I don't know if you can see that. So this is where your physiological buffer systems come in, your respiratory and your renal systems. So they are slower, right? Chemistry happens really quick, but it takes a while for you to exhale a whole bunch of CO2 to lower your carbonic acid levels. But because you're breathing in and out all day, all your life, and your kidneys can pump and pump and pump hydrogen ions or bicarbonate ions, they have a larger capacity. So they kick in when your chemical buffer systems are being overwhelmed and can, over time, bring your pH back down where you want it to be. So just to review, and we have the Physio EX due about this week. I don't know when you're listening to this relative to that. Touches on this. So you just want to review how your kidney is going to respond to changes in pH. If your blood pH is too low, this means you have too many hydrogen ions in your bloodstream. So what your kidneys are going to do is make sure that the hydrogen ions end up in the urine and bicarbonate stays in the blood. So you're going to have low bicarbonate in the urine, high hydrogen ions in the urine. And then those hydrogen ions are going to be neutralized by the phosphate so that your urine doesn't get too acidic. If your blood pH is high, if you're becoming a little bit alkaline, then what your kidneys are going to do is secrete bicarbonate into the urine and retain hydrogen ions in the bloodstream. I just noticed that was missing, so I paused, put it in, and then started recording again. So that probably just magically appeared for you. Make sure you write that into your notes. So if your blood pH is high, too alkaline, dump bicarbonate into the urine, hold on to all of your hydrogen ions, blood pH comes back down. Last two slides. Maybe one or two questions at most about this on the exam. So we already said before that for the most part, your metabolism tends to acidify your bloodstream. Under normal circumstances, your metabolism doesn't really cause acidosis or alkalosis because you have these buffering systems to keep it stable. If you lose your homeostasis for some reason, then you can get metabolic acidosis and metabolic alkalosis. So in the case of too much alcohol ingestion, when the alcohol gets broken down, it gets turned into acetic acid, which is very similar to lactic acid. They're both lactic acid and alcohol are the byproducts of incomplete combustion, so they're chemically similar to one another. Diarrhea can cause excessive loss of bicarbonate. So what happens is your digestive tract dumps bicarbonate into the lumen of your digestive system to neutralize all of your stomach acid. And if you keep emptying the contents of your intestines, then you're flushing all of this bicarbonate out of your system. There are hydrogen ions left behind from the production of that bicarbonate, because what your body actually does is manufacture carbonic acid, and it dumps the bicarbonate into the lumen of your intestine and leaves that free hydrogen ion floating around inside your body, which then has to be neutralized some way. But if you're dumping lots and lots of bicarbonate into your intestine because you keep emptying it, then that leftover hydrogen ion ends up causing your blood pH to go down. And then these other things like a lot of lactic acid, but this would be like extreme exercise, which I won't go into details on that. I'll just move on. But be familiar with that. And then metabolic alkalosis, as this says, is much less common because your metabolism in general tends to acidify your bloodstream. The best example I have of metabolic alkalosis is caused by the vomiting of your stomach contents. So it's the reverse of what happens in your small intestine. So technically what ends up in your stomach is hydrochloric acid. But what actually happens is the cells that line your stomach make carbonic acid and dump the hydrogen ion along with a free-floating chloride ion into your stomach. So what your stomach has inside of it is hydrogen and chloride, or hydrochloric acid, but it's actually a combination of carbonic acid plus the chloride. So every time you empty your stomach contents and get rid of all of that stomach acid, what your stomach has to do is make more carbonic acid, dump the acid into your stomach, and then that bicarbonate is still floating around. And if you get an excess of bicarbonate from your stomach, then that gets into your bloodstream and lowers your blood pH. So that's it, 28 minutes for that chapter, relatively straightforward. Thanks for sticking around to the end.